Hello, everyone. Welcome to our live interview series for Cascades Analytics. My name is Alexandre Stepanovic. I'm your host and founder of Cascades Analytics, a media and consultancy bureau based here in New York City, specialized in psychoplastogens and psychedelics. And today I am really excited to welcome the Senior Director of Preclinical at Beckley Psychtech, Amir Lotfi. Thank you, Amir, for joining us from Oxford, the UK. How are you
Yes,
doing?
that's great. Hi, Alexander. Yes, I'm doing very well. I hope you are too. Thank you for inviting me on this talk. Pleasure to be here, and welcome to your audience as well.
Yes, thank you, thank you. So we were just talking before the live about how you heard about psychedelics, and it's kind of a full circle for you to be working there. Could you tell us a little bit your story about how you first became interested in psychedelics?
Yeah, absolutely. This takes me back about almost 15 years now. I got introduced to psychedelics from various sources, but I think the most important one of them was Amanda Fielding's work and her activity at Beckley Foundation, which is very very inspiring, and the honesty and the passion in her, in continuing that activity was very and captivating to myself personally. And obviously at Beckley Psychtech, we're taking that legacy and her activities for the past 25, 30 years. And trying to make these products and medicines into accessible format, into a regulatory framework. So yeah, initially when I started kind of delving into the psychedelic literature, I never thought that there would be a time and place where we're all working on psychedelics and there's going to be companies developing these medicines. So yeah, it's very it's very humbling experience to have been part of this process. Yeah.
Yeah, were you firstly interested in 5-MeO-DMT? I mean, did you was this like your psychedelic of interest at first, or did Beckley Psychtech drive you there?
It wasn't my own kind of interest. I knew about the better known DMT, and I knew there was a more vague analog of that called 5-MeO-DMT. But I mostly got introduced into the psychedelic space with the better known compounds, LSD and psilocybin, and that was the start of kind of my own interest and studies. But yeah, talking to people in the early days of Beckley Psychtech, my direct manager Tim Mason, we were kind of discussing the different compounds, and 5-MeO-DMT was very much in the forefront of the discussions, and also Amanda Fielding herself had an interest in 5-MeO-DMT alongside LSD and psilocybin, which were mainly I guess mostly were her interests. But yeah, essentially 5-MeO-DMT because of the reliability in inducing the psychedelic experience, as well as it being a shorter duration of action, which makes the scalability and the in clinic supervision a little bit easier compared to other psychedelics. Those were the reasons that we chose to work with 5-MeO-DMT.
Mm-hmm. Mm-hmm. And so, how does your preclinical program at Beckley Psychtech help de-risk compounds before they move into clinical trials?
Yeah, so specifically for 5-MeO-DMT, it was very novel kind of approach because we wanted to do an intranasal route of administration, and it's not that many compounds or CROs that have had experience with this at the time that we were looking. So we basically tried to obviously formulate a novel salt form of 5-MeO-DMT, and we used some some of the well-known excipients, to basically design and formulate the early product of 5-MeO-DMT, which we used in animal studies, as well as the first clinical trial with 5-MeO-DMT at Beckley Psychtech. We basically took 5-MeO-DMT intranasally, and we performed most of the conventional toxicology work, safety pharmacology and toxicology and drug interactions, in order to obviously profile and conceptualize the adverse events and safety profile of it, and make sure the risk benefit is there before we move into the clinic. And yeah, that's one aspect. And then the other aspect of it is obviously understanding the PK profile, understanding the drug interactions, as well as the whole host of requirements for taking a compound into first-in-human trials.
Because nobody was working really on 5-MeO-DMT back then, so you had to do everything from scratch, or?
Most of the things that we did was from the scratch. Absolutely. I mean, we used a little bit of epidemiological knowledge that's out there, but I believe most of that is from an inhalation route of administration, which wasn't exactly what we were planning to do. There is some information about intranasal, but we mostly had to start from scratch and develop our own product and conceptualize and profile the safety, safety of 5-MeO-DMT ourselves, because it was a novel salt form, not novel excipients, but novel excipients in this formulation, which obviously has an impact on the exposure and on the time course of experience. So yeah, we basically did all of those aspects from scratch. Yeah.
And how do you test in rodents an intranasal? I mean, this is quite peculiar.
Yeah, we had to do a little bit of like creativity. We worked with a little bit of excipients, and we modeled. There was some change in the clinical product because it's using a device, which has an impact. But in the rodent studies, we opted for like a pipette installation, basically in the nose, and we had a good, decent exposure, which allowed us to assess the systemic and local toxicity of this compound in rodents. But obviously, we also used a second species, which were dogs in our case, and we managed to use the clinical product. So yeah.
So yeah, there are both sniffers, huh? The rodents and dogs. So I guess that wasn't too hard. But so including behavioral animal models, what are the translational models that you found most useful for developing psychedelic-based therapies?
Yeah, that's a very good question. I mean, it's an age-old discussion, isn't it, in neuroscience and like translational models. I'm gonna reply to that in two kind of sections: one for 5-MeO-DMT and one for our other earlier stage assets, which we can talk later in this discussion. But for 5-MeO-DMT, we essentially used synaptogenesis in vitro and in vivo synaptogenesis, just to understand some of the maintenance of effects from a single dose. We used some of the behavioral models, such as learned helplessness and things along those lines, but we've stopped that. That was, historical studies, and we haven't really, continued with those behavioral models. There are some limitations in translation of those models, specifically for psychedelics. So yeah, those are some of the models that we use for efficacy of 5-MeO-DMT. We also collaborated with some other academic as well as industry CROs to further assess the profile, the safety and toxicology, as well as efficacy of 5-MeO-DMT. But obviously, a lot of the information about the efficacy of 5-MeO-DMT is derived from epidemiological studies, and obviously our own clinical data, the recently read out phase 2b data, which shows a really good efficacy with 5-MeO-DMT.
And what about the head twitch response? I mean, in 5-MeO-DMT, that must be tricky because the hallucinations are not as strong, although the correlation between head twitch and hallucinations are not always direct. So what do you...
Yeah, exactly. I mean, we did do head twitch response with 5-MeO-DMT, but it was mainly to compare with the library of new chemical entities that we were working on at the time. As you said, hallucinations, or maybe I should say sensory alterations with 5-MeO-DMT is not as strong as it is with some of the other well-known psychedelics. And I want to be careful, but I might think that head twitch responses might not be the best model for capturing the nuanced differences between maybe 5-MeO-DMT and psilocybin and other psychedelics. But we did do head twitch response, and we had a good kind of we had a good response with our model.
Oh, okay.
I mean, we did a dedicated like head twitch response with subcutaneous route of administrations, but then also captured this response in our toxicology studies where we used an intranasal route of administration.
And do you use any in silico tools like AI, machine learning, or predictive modeling?
Yeah, absolutely. For regulatory studies in 5-MeO-DMT, we used a structure-activity relationship kind of predictive tool. They're called direct and lead scope. They were to assess, any red flags associated, about mutagenicity and carcinogenicity, and there's a range of endpoints in that tool that you can assess, various endpoints. And we also used, PK predictive modeling in order to predict those levels for our human escalation designs. So those are some of the things that we did for 5-MeO-DMT. But for our NCEs, a little bit more extensive. We used molecular dynamics, modeling. We used some of the structural binding, kind of computational modeling. This was done with with a computational chemist that we've been working with. So yeah, those are some of the things that we did using AI. Yeah. Or yeah.
Yeah.
But it's also kind of a personal interest for me as well. I know NAM's new approach methodologies is very much a hot topic these days. And I think 5-MeO-DMT might be a little bit late stage for us to be using those kinds of methodologies, but it's something of a personal interest, and I will be using those approaches in the future.
Wasn't it a lot of pressure on you to study preclinical effects when you already knew that it had, such a strong impact on humans, and trying to replicate this on level of synapses or behavioral models?
Yeah, absolutely. I mean, it's it's like it's always been a question for myself, like, what is it with psychedelics that we're trying to assess in preclinical models, obviously, other than safety and efficacy and what's needed for moving a product into clinical development. But I mean, the answer to that is, I'm still asking the question, but the answer for me is we're essentially trying to model some of the components in non-clinical. We're never going to be capturing, the deep psychedelic experience in a simplified animal model, and with our own kind of methodological limitations that we have. But we study components, right? Synaptogenesis, neuroimaging, electrophysiology, and stuff like that. And for me, the reason for that is I guess, to come up, with new molecules and with kind of being selective in the components that we want to carry forward in any particular kind of drug development project. But yeah, it's a very interesting, kind of field to be in, because the information is out there in humans. So what exactly are we trying to back translate in animal models is always a question for me.
So could we dive a little bit further, deeper into the BPL-003, and for those unfamiliar, could you tell us what it is?
Yeah, sure. So 5-MeO-DMT, or BPL-003, is an intranasal formulation of a novel salt form of 5-MeO-DMT. It's a 5-MeO-DMT benzoate, which is formulated with a few inactive ingredients to make them conducive for an intranasal administration. In terms of what 5-MeO-DMT is, it's an alkaloid. It's naturally derived. It's observed in several plants and animals. Probably the most famous of which is the Colorado river toad, which has been historically used for sourcing 5-MeO-DMT. But we obviously make our own 5-MeO-DMT benzoate in the lab. It's a synthetic molecule. So from that aspect, maybe some of the other minor alkaloids or some of the other compounds that might be present in the bufo toad might not be or not present, obviously, in our preparations. But we obviously have a control over the ingredients that go into the human trials.
And how often is BPL-003 described as, it is described as the next generation 5-MeO-DMT analog, but so you said it differs from natural 5-MeO-DMT, but what does that mean in terms of safety and therapeutic potential? If you could tell us a little bit about that.
Yeah, sure. As I mentioned, the naturalistic 5-MeO-DMT is used mostly from an inhalation route of administration, which is not exactly a accepted medical kind of route. So we chose an intranasal route of administration, which means that you have to kind of assess the safety from this route of administration, make sure that local tolerance and things like that are also profiled extensively. So from that aspect, it differs, from the naturalistic 5-MeO-DMT. From a therapeutic perspective, we haven't kind of done head-to-head studies with naturalistic, 5-MeO-DMT, so I can't maybe make a comment a lot about, the efficacy of naturalistic. But there are obviously epidemiological reports out there that puts naturalistic 5-MeO-DMT somewhat efficacious in various various conditions. But again, our BPL-003 has been in the phase 2b. We've profiled the adverse events, which are mostly, I want to say, up to 99% have been mild to moderate. So the safety profile of it is very favorable. And in terms of maybe another aspect of the experience is we're going for an intranasal administration. And one of the concerns that we had in the early days, and this again comes from some of the epidemiological reports, that there is a nasal irritation with naturalistic 5-MeO, whether it's freebase or some of the other common salts. And we actually tried to kind of screen in a very basic model of nasal irritation, and we came up with this benzoate salt, which had a very low potential for causing nasal irritation. So we also tried to optimize the experience from that aspect as well.
Is it the same intranasal spray as Spravato? Is it the same mechanism, or?
I am not sure about the kind of the device that Spravato uses. So I can't comment on that.
Sure. So 5-MeO-DMT is known for very intense and often non-visual, near-death-like experiences, and some find it transformative, and other might find it overwhelming. So, from a translational or clinical perspective, what makes it compelling enough to study despite those challenges? And how do you balance potency with patient safety? Is there like a screening? Is there like a pre, a screening of sorts?
Yeah, absolutely. I mean, it can be a very overwhelming experience. But at the same time, as we understand the safety, the physiological safety of 5-MeO-DMT or BPL-003, we understand most of the kind of overwhelming nature comes from, psychological, impacts of 5-MeO-DMT. And, just drawing from the literature and again, what we understand from other psychedelic compounds, even what can be overwhelming for some participants can also be therapeutic. Even those challenges might have some learnings in the end for a participant. But at the same time, we make sure that all the participants who are coming to our trials are safe, are screened properly. If there's any signal from a preclinical perspective, we make sure that we monitor that in the clinic. We made sure we obviously understand the interactions with other drugs, drug interactions and things like that. And we made sure that we exclude, patients, based on justifications and based on the data that we have. Yeah, but at the end of the day, we understand that as the saying goes in toxicology, the dose makes the poison. So we understand that we have a good margin of safety, and despite the nature of the experience, we understand the physiological safety of this compound.
Are you looking for a second dose? I think I've read recently that you're developing like a second dose approach protocol.
Yeah, so the open label extension of our phase 2, a study with 2 doses has just reported out, I think with an 8 mg followed by 12 mg. That was the model in our in our previous clinical studies. I believe there is another readout in a few weeks' time as well, which we're all looking forward to excitingly. But yes, that is the current model. I mean, it is being developed based on the data that's just read out, and the data in the next few weeks. Those will impact how the phase 3 clinical protocol will be developed, and how the trial will look like in the future.
So based on publicly disclosed data, what have we learned so far about BPL-003, both preclinically and clinically?
Yeah, so from a clinical standpoint, I mean, it's got a very nice efficacy response. Obviously, from a single dose, it is extended to almost 2 months. I mean, the second dose seems to even drive the efficacy signal even further. So that's a very positive thing. From a safety standpoint, obviously, as I said, most of the adverse events have been in the mild to moderate kind of category. It's very well tolerated in the patients, in the patient population that we're working with. So yeah, it's a very kind of exciting time, and we're looking forward to hopefully our phase 3 approval and trial. From a non-clinical perspective, again, since we've identified the risk profile, and obviously it's been in front of a lot of regulatory agencies, it looks like the risk benefit is very favorable to take this compound to clinical development. Yeah.
Yes, it's fascinating. It's not the only product that Beckley Psychtech is developing right now, but it is, I mean, the one that's super promising today, and that gets all the attention. I don't know if you want to say a couple of words about the other ones, or
Yeah, I just wanted to kind of raise another indication for 5-MeO-DMT.
Yeah.
We also worked with alcohol, in a smaller trial with alcohol use disorder, where a single dose of 5-MeO-DMT drove, or BPL-003 drove the heavy drinking days to a very significant degree, and this is all very, very exciting as well for a second indication for 5-MeO-DMT.
Mm.
Yes, we also had a second product called LE101, which is a psilocin formulation, an intravenous psilocin formulation, which is, I believe, in phase 2A in the UK. And yeah, we're going through various milestones in Europe as well as the US in the next few months.
Great. Does Beckley Psychtech collaborate with academic or external partners? And could you share a few examples?
Yes, absolutely. I mean, in the clinical space, we're working very closely with Imperial College. I mean, they're one of our partners in driving the clinical studies forward. They're great researchers and PIs in this program.
fMRI and DMT program?
There was an EEG study. Yes, there was an fMRI study as well. Yeah, exactly.
Yeah.
That's our main kind of clinical collaboration. We've also worked with a lot of industry partners in the preclinical space, as well as some collaborations with academia. Providing 5-MeO-DMT, kind of discussing the projects and things of that nature. Yeah, some nice collaborations.
Looking ahead, what excites you most about the future of translational neuroscience in this field?
I think there's a lot to look forward to. I mean, psychedelics, classical psychedelics, I think will have a really good space, good position in psychiatry. I'm very much looking forward to the pipelines in the many companies and how it will look like in the market, hopefully in the next few years. A little bit more translationally, I'm very much interested in understanding how these new approach methodologies will complement or replace animal models for safety assessment. That's some of the things that I'm looking forward to. I mean, there's obviously a lot of medicinal chemistry activities being done on psychedelics and on psychedelic derived compounds, which I'm personally very interested to see how it looks in indications other than psychiatry, inflammation and neurodegeneration and even things like pain and chronic pain are, very much of my own personal interest. And I'm very much looking forward to see how the space will unfold.
All right. Amir, thank you so much for your time today. That was very enlightening. Thank you. I wish you the best at Beckley Psychtech, and hopefully, I can see you at one of these conferences around the world.
Great.
And well, thank you for your time today.
Absolutely. Thanks a lot for having me on, Alex. Really appreciate it. I enjoyed this talk. Yeah.
Yeah, me too. Thank you so much. Thank you, everyone, for tuning in. Don't hesitate to reach out if you have any question. I can forward those to Amir, or if you need the transcript of this conversation. And I'll see you soon. Thanks for tuning in again, and bye.